Perfect. Okay. Hi, I'm your session chair for this session on mid-throughput sequencers in 2024. This is kind of a follow-up from last year's. If you were in Boston, there was a thing on the same kind of instruments, and now since it's been a year, and there are a lot more in the field, I was hoping that we could kind of, we have some expert users, and just some questions, so please think of any questions you have for the instruments that we'll be talking about today. But first, we are going to start with a sponsor talk from Singular, and that's from Darius, and then we have a lightning talk from Hongduan from BD. So they'll happen first, and then we'll get started with the sequencers talk.
Thank you. All right. Good to see some familiar faces in the audience. I'm Darius Fugere, lead up marketing and product management at Singular Genomics. We'll be talking about current and future applications of G4. So for those of you that don't know, Singular Genomics was founded in 2016. We're based in San Diego, California. And that's where we do all of our research, development, and manufacturing. Really from the beginning, the team's been an innovation engine, now over 260 issued and pending patents. And we're in a strong financial position with over 170 million on the balance sheet as of our last earnings call. And really our mission right now is twofold. One, it's to support the G4 sequencing platform and our customer base, and launch the G4X spatial sequencer, which I'll talk through. If you're not familiar with the G4, this is a fast, versatile, and powerful benchtop system. It's all runtimes under 24 hours to support all types of applications. It's got the power to produce 1.6 billion reads per day, and that's split across 4 flow cell positions. Each of those flow cells has 4 fluidically independent lanes, so you can get 50 or 100 million reads per lane across 16 lanes per run. And then kind of gold standard accuracy with 85% of bases greater than Q30. And that's really enabled from our novel 4-color SBS chemistry that was developed in-house. So our G4 is really compatible across the gamut of applications. Our growing customer base in the field is using it for a variety of different purposes. And the company's really been thoughtful about partnering with the leading library preparation solutions providers to enable the G4 to fit within existing lab ecosystems. And all of this data we've produced, and it's available as application notes on our website if you want to learn more. So what's coming next for sequencing on the G4? So actually starting about 4 or 5 years ago, Drew, Eli, and the team started thinking, where can sequencing be particularly disruptive within that application ecosystem? And it became clear that in situ sequencing is something that was going to be kind of the next frontier for sequencing. So the question really became, could we enable a spatial on the G4? So if you kind of took the covers off of the G4, what you'd find is we've got NVIDIA GPUs and AI-powered base calling, and the processing power to analyze over 2 billion pixels per second. We have an advanced fluidic system to accommodate the 4 flow cells and 16 lanes. We have a ultra high speed imaging system with 0.5 micron resolution camera. And we have expansive imaging area across those 4 flow cell positions. And last but very importantly, we have precision heating and microfluidics that can be adapted for advanced spatial protocols within the nest. So the answer is yes, we can. And at AGBT just a few months ago, we introduced the G4X spatial sequencer. This is upgradable from the G4 and allows it to be used for ultra high throughput spatial sequencing. So instead of loading your NGS libraries onto the flow cell, we're actually working directly from tissue. So you can put the tissue on the flow cell, and we have a unique methodology. You can stop by the booth and see how we do that. And then when you press start on the system, it's going to produce multi-omic data on that tissue sample. So starting at the bottom, we produce an automatic fluorescent H&E. This is really nice. You can visualize the tissue morphology and connect into your standard histology or digital pathology programs. We produce proteomic readouts, so you can see dozens of proteins that are imaged at high resolution. We do transcriptomics. This is a barcode-free profiling of hundreds of gene transcripts with high sensitivity and specificity. And then a very powerful methodology called direct-seq. This is where we're actually sequencing a variable region of RNA. Think B or T cell receptor sequences of the CDR3 sequence, cancer hotspot mutations, gene edits. And we can do this all together on the same section, an FFPE section, and we can do this at subcellular resolution. So taking a quick step back I think that's a powerful multimodal readout that the field can benefit from. But one of the biggest issues in spatial biology is really that right now it's severely limited by throughput of systems and the price per sample of running spatial experiments. And we look at moving forward, researchers want to run more samples. They want larger studies with more power, especially as this starts moving into translational and clinical research. And they also want to do more sections per sample. They want complete 3D reconstruction of the tissue for a better understanding of the underlying biology. So really, tomorrow's studies are going to be more sections across more samples, and this is an exponential issue that we see moving forward. So the G4 is addressed to solve this problem, enabling multi-omics spatial biology at scale. So the leading in situ platform, you can fit about 4 samples per run, and you can run that about 3 times a week. With the G4X, we have 10 times the imaging area, and it's twice as fast. You can analyze over 20 times the number of samples per week on the system. So this is really powerful and should unlock a huge amount of studies moving forward. So this is really 2 technologies, 1 singular platform. The G4X is capable of all of the next generation sequencing aspects of the G4, fast, flexible, powerful sequencing. And that's really nice for single cell sequencing, ex situ transcriptomic studies. And then you can also do spatial sequencing, so all your in situ work that I talked about, 300-plex RNA, 15-plex protein, all in a daily run. So what's next for Singular? Supporting our next generation sequencing moving forward. We have a V2 upgrade coming forward with some new software functionality and improved reliability and robustness with some hardware upgrades. We have Singular Connect, which is also enabled in that update. That is really nice off-instrument results visualization, live run monitoring, user management. And then our F4 flow cell, which we expect to produce between 600 and 800 million reads, which can potentially double the output of the sequencer. And then we're also developing the spatial sequencing part. So right now we're running these internally through our technology access services. We actually have a research grant, a $50,000 research grant that's open for another week and a half, I believe. So a great way to get involved there. And then towards the end of the year, we'll start translating this technology into a couple key early access sites with broader commercialization happening next year. So that's kind of it. Really encourage everyone to get connected. You can stop by booth 216, learn more about the G4 or the G4X, see the tissue transfer methodology I mentioned. We've got tacos and tequila tonight at Sonora Mexican restaurant, which is pretty close. And then we have a Wednesday morning breakfast workshop where you'll get an hour deep dive into both the sequencing and spatial components of the G4X. Thanks.
Good afternoon, everyone. My name is Hongduan Huang from BD Biosciences. Today, I will be introducing our newly developed microwell-based single-cell multi-omic ATAC-seq and RNA-seq assay, which allows users to study the chromatin accessibility and the whole transcriptome at the same time. So our standard multi-omic single-cell ATAC assay uses the nuclei as the input sample type. After nuclear isolation and the in situ segmentation with TN5 transporases, those nuclei are loaded onto our BD Rhapsody HD Express station. On those 8-lane cartridges, the single-cell partitioning is achieved within those microwells. After lysis, the ATAC DNA fragments and the mRNA are captured by our Rhapsody beads. Following the library preparation and the sequencing, thousands of unique ATAC fragments enriched at the transcription start sites are obtained by our assay. And those ATAC fragments show high correlation with the RNA molecules from the same cell. So one of the most attractive features of our multi-omic ATAC assay is its high flexibility on the sample type. Robust assay performance has been demonstrated on various cell types, from cell lines, tissues, to fresh and frozen PBMCs, and with a very wide range of nuclei input. From as low as 1,000 nuclei per lane to as high as 400,000 nuclei per experiment. And more importantly, our assay can not only work on the nuclear samples, but also works for the whole cells. So in this case, you don't need to isolate the nuclei, and the assay performance is very comparable to the nuclear ATAC. So if for your specific research interest, you wanted to use the whole cell as the input sample, we can work together to accommodate that. And in addition to the flexibility on the sample type, our workflow is also very flexible. Our Rhapsody beads bearing the ATAC fragments and the cDNA from the mRNA can be stored at 4C for up to 4 months without changes on the assay performance or the biology. So users can freely determine their timeline of experiments and also the number of the cells they want to subsample and sequence. And most excitingly, our multi-omic ATAC assay is compatible with our nuclear sample multiplexing tags. So different nuclei can be tagged differently, then put together to load on one lane. Then we can conduct the experiment altogether for different nuclei, which can increase the throughput of the experiments and also reduce the cost. So overall, our robust and flexible multi-omic ATAC assay can serve as a powerful and reliable tool for the studies in epigenetics. Thank you, everyone. So if you have any questions, please stop by our booth at 405 and feel free to reach out to us. Thank you.
Stopping by today. And thank you to Molly for the invite to give this talk. Today I'll be talking about what we've learned with our Singular Genomics G4. I'm at the University of Vermont, part of the Vermont Integrative Genomics Resource. Just a little background on myself. I've actually only been in a core facility for like 2 years, so I haven't had much experience with like past sequencers. As far as like what we do as a facility, I think we're considered kind of like a mid-throughput genomics core. Our fearless leader is the infamous Scott Tye. I hear some chuckles in the room. I think some people might know him. We have a couple of senior techs as well as junior tech and some wonderful undergraduate interns. As far as what we have for sequencers in the lab, we have our trusty old MySeq. We have the G4 that we'll be talking about today. We also have our GridION. We're big fans of Oxford Nanopore in the lab, as well as our brand new P2 Solo. So how did we end up with a G4 in our lab? Well, previous to that, our kind of workhorse in the lab was our old HiSeq 1500. I think the core purchased it in like 2011-ish. Yeah.
So this was the first instrument that I ever sequenced on because I did join the Corps 2 years ago. And it was quite the experience, let me tell you. Anyways, so starting around fall of 2022, we kind of said, hey, we need to sunset this instrument. It's end of life. I believe February of 2023. We bought our last reagents in December of 2022. So in the fall of 2022, at that point, it was kind of like, okay, I guess we'll get an SC 2000, right? But then we took a long time to make a decision, and all of a sudden, we had decisions to make, right? So it was then, do we get an XSeq 2000? Do we get an element of VD? Do we get a singular G4? And so a lot of considerations went into how we made this decision. And I'm not going to go through our rubric or anything. This is just to show all the things that we took into consideration when buying this instrument, because for us, we're not a huge genomics core, so we pretty much have like one or maybe two benchtop sequencers, right? So we're really relying on our instruments. So in the end, what it came down to, each company definitely has their strengths and each company has their weaknesses in all of these different categories. And some of those have changed since a year ago when we were comparing them and doing our shopping. But what it came down for us, really and truly, was the flexibility of the system. And why we prioritized that was we have investigators who are sequencing kind of crazy stuff all the time. We're not just human and mouse. We're jumping worms. We've got this spider lady. We have this rather large fish project with the state of Vermont. We get Drosophila. We get a lot of plant. And as people probably know, Scott's involved with extreme microbiome. So we're always getting really strange things from that. So on the system as well, we've gotten all sorts of different kinds of libraries that we've used on our singular so far. So this is just a whole suite of different library preps that we have sequenced. For the most part, our users just they don't want to change their library preps. So they don't have to. They send them to us. We convert them to be, so that they can bind to the singular flow cells, and then we just sequence them. We have developed a few de novo preps. We mostly use the Revity Rapid XP in our lab for DNA sequencing. And Scott's been working with Sequence to develop a de novo prep on their RhinoSeq DNA-seq library. And we've also gone and developed a de novo prep for the nanostring GMX, which we have next door in microscopy. So just a few quick stats on what we usually see for output. We have two flow cell flavors that we really run, the F2 and the F3. For the F2, that's the smaller flow cell. We're usually seeing between 215 and 300 million reads for output that are passing filter. The average is 265. For the F3, larger flow cell, 345. A couple of times we've hit 500, a little bit above, with the average being 400. And then down here, so one of the joys of working with the HiSeq was the art of the loading concentration. We spent so much time saying, oh, should we load 17 picomole or 17.5, right? And this was a whole discussion in our lab. And so what we really like about the pattern flow cells on the G4 is we don't have to do that really very much. Usually it's, hey, put in 25 picomole for an F3 and run it and go, and we'll get plenty of data out of it. We did run one loading concentration titration on a library where we loaded 25 picomole and it just was not hitting what we were expecting, and it turned out it's a larger library, larger fragment size, didn't bind so well. So we loaded 40, 50, 60, and 70 picomole. And you do see some drop off in the Q scores towards the end. especially with the higher, like the 60 and 70. But we're still getting the same number of reads at the end that are passing filter. So, and then real quick here at the end, we've been working a lot in our lab on adapting library preps to the G4. And so we did a quick assessment, and I mean quick, like we just got some of the FASTQs in on Friday. So this is very last minute data. It's not fully analyzed yet. But we ran the Revity NextFlex Rapid XP, the Watchmaker DNA library kit with enzymatic fragmentation, Takara Throughplex, and NEB's Ultra Express. And what we're also looking for is streamlining things in our lab and getting as little hands-on time as possible, right? So the DNA inputs that we used for this study, we used some just human genomic DNA. We also used the MSA 1000 standard from ATCC, as well as Scott's dog, Maggie, some of her genomic DNA. So just a few stats on these different kits. We used 5 nanograms of input across the board. For the NEB kit, this was kind of outside of their recommended input amounts, just a little low. I think that's a 10 to something. I can't remember what the high end was because we don't usually go with high end stuff. PCR cycles, all pretty similar. And then hands-on time, the Takara and the NEB are a single B cleanup at the end, so less hands-on time. Whereas Watchmaker and Revity have a B cleanup after adapter ligation. So as far as library size distribution went, they're all pretty consistent within, so like the 5 different preps that we did for a kit, right? We definitely didn't have time to like optimize our fragmentation time or enzyme concentrations to get like perfect things, right? This was just a quick and dirty experiment. So I think we would do some of that, particularly with the Watchmaker and NEB. We were just following recommendations at the time. And then finally, we got FASTQs back on Friday, so I just threw stuff through FastQC just so that I could show some quality scores. So in purple, we have Watchmaker, Takara is in blue, Revity's in green, and the NEB kit is in orange. They're all fairly similar. Everything is pretty much over Q30 for most of the run. And what we would like to be able to show, it's coming up probably next week, is we're going to look at what we're pulling out of that ATC 1000, because that's 10 different bacterial strains or microbial strains. And so we want to see if we're getting close to the distribution in that. So with that, I'd like to thank you for your attention, as well as I'd like to thank the folks that I work with and the support that we got from the various vendors for that last little project.
Thank you so much for being here at this time when you are just looking forward for your drink and you're still hearing about some science. So I know that how it feels. And thank you, Molly, for this invitation. So my name is Boni. I'm director at Yale Center for Genome Analysis. And we got this instrument pretty recently. And so this is our lab. It's around like 11,000 square feet facility with 35 people working in our group. We have a high-performance computing system to analyze the data, and we have like I have shown some 6,000, NovoSeq 6,000, but we also have a assortment of sequences. And we approximately produce around 1 petabases of data a year, which is approximately, say, 90 petabases per month. So we are a very high throughput facility. So then the question you will ask, like, then why you need a mid-throughput sequencer? You have all these gizmos, and then why you want to buy one. And so that is where actually I would like to draw your attention towards a use of a mid-throughput sequencer, especially a versatile systems like AVT can actually help you. And before joining Yale, I was in University of North Dakota, and the lab that I was actually working. It was a very low throughput lab. And I always wished like I had something like a high-seq in my group. So that is the void that AVT, or even the singular actually, what Krishna was talking about, fills that void. And so this instrument is amazing. This is actually, can be used for multiple things. But before going into that, this is an assortment of services that actually we provide from YCGS. So you can see like all different kinds of libraries we are producing within the facility. And so that will find a home in one or other of this instrument. So to just introduce you to this platform, if you are not aware about it, it actually produces data, pretty good data actually. We consistently see data produce more than 95% of data coming with a Q30 value. And almost 80 to 85% data is actually at Q40 ranges. And actually, if you are running at 150 base pair run rates, then quality don't drop at the end of the run. So that is actually the pretty amazing aspect about this sequencer. But the question is, like, do you really need that high quality reads for all the applications? The probably answer to that question is that no. So many of the applications like ATAC-seq, ChIP-seq, or RNA-seq, if you are doing, you really don't need a Q40 reads. So when you are calling for a variant or those kind of analysis, then probably the Q40 reads have an application. So when you are thinking about read quality and making your decisions based on whether a sequencer can produce a Q40 or Q30 reads or not, you have to think about it, whether your experiment really need Q40 reads or not. But when it comes to cost, the cost of sequencing in AVT is almost equivalent to NovaSeq 6000. So it's a sequencer which is a mid-throughput sequencer, but the cost economy actually works almost comparable with the NovaSeq 6000 range. It has two fully independent flow cells, so you can actually sequence two different set of samples, and each of them have two lanes. So you can and those are also independent lanes. And the highest throughput flow cell will give you around 1 billion reads per flow cell. The other beauty of this instrument, it has multiple options for you to run an experiment. Like you have a low throughput option, you have a medium, and you have a high. So if somebody wants only 200 million reads, then you can actually choose an appropriate flow cell to run those samples, rather than to run a 1 billion flow cell and charge them for 1 billion. Or tell them that, oh, you need only 200 million reads. Either you wait for 5 more person to come and give their samples, or you have to you have to take the cost. So you can pick and choose your flow cell, and then based on your need, and that also gives certain amount of economy in terms of your run. And also it will actually allow you to get to manage the turnaround expectations. Like one of the most important applications that we are using AVT is for sequencing user prep library. So in our facility, we are preparing around 50,000 libraries a year, but we also get around 10,000 libraries from the users. They prep those libraries and they submit us for sequencing. So those libraries, sometimes they are coming and just tell us, hey, I need this tomorrow because there is a grant deadline is there, or a paper is waiting, and can you run? And so our model is like we bulk all our libraries and then run them on a NovaSeq X Plus kind of a system. So that means they have to wait. They have to wait at least for a week to make those logistics work. So in this scenario, we can say, hey, we have a 300 million reads. If that is what you want, then why don't you use this flow cell, and we will give you data in another 3 days or 4 days. So you can manage, based on this flow cell availability, you can manage expectations for regarding the turnaround time, especially those who are looking for rapid turnaround for various reasons. And this is kind of a cost comparison with various platforms available. At this point, NextSeq 2000 is somewhere between AVT low and mid throughput flow cell. But with this new XLEAP chemistry, it's going to fall the price will fall somewhere between mid and high plex flow cell. So basically, a little bit, NextSeq 2000 will be actually reduced, price will be reduced with the XLEAP chemistry, and then the position of that dot for the NextSeq 2000 will come somewhere between AVT mid to 150 and high 150. So still, the high plex flow cell in AVT is cheaper than NextSeq 2000 flow cell. So this is, I'm just talking about the list price. So people have their own arrangement with individual companies where they get some extra discounts and which is not taken into account in this. So ABRF works based on a amplification mechanism called as rolling circle amplification. And the advantage of using rolling circle amplification is that a PCR error, which is called as scar-based errors, are not actually propagated. So what happens in your normal PCR, that you amplify, and whatever products are actually generated, they are amplified in next generation. So if one error is introduced, it gets amplified over number of cycles that you are, and that takes place in during the cluster generation step. So when you are doing rolling circle amplification, the amplification takes off always from the template. So during the amplification, if one error is introduced, that error is not propagated because actually, again, the template is used for amplification. So this gives it pretty, that's where some of the advantage of higher read quality comes in the ABRF. And again, it actually uses multivalent avidites. This is basically you are accumulating the signal, a low intensity signal, and making it darker with a high signal to noise ratio using this multivalent binding. So that also gives you a better base quality when you are sequencing. There are two different modes are possible for ABRF. One is called as expert mode, and another is called as conventional mode. Conventional mode means like whatever I have shown you, like a billion read or something you are getting, you will get that 1 billion reads. But if you run them on an expert mode, that instrument, and you don't have to do anything, especially you don't have to buy anything, just change to expert mode, and you start getting extra data. The question is that where that free data is coming from. Like you see that ad in the television that free money is there kind of stuff. So where that free money is coming or free data is coming. So what happens that once you run them on an expert mode, they start reporting those polygons or those sequences which are slightly lower quality. So you will get those data also, and then an informatician can go inside the data and then can trim and then remove anything that is not acceptable. So that way, from a given flow cell, you can increase your output to a higher. And we have run our instrument in both the settings, and actually it works pretty well. One thing I should tell you that expert mode, the yield is always variable. You cannot claim that this is the yield for that. So if you have 1 billion flow cell you're running, then you can always say like 1 billion flow cell will have this much yield. But in expert mode, it can give you 1.3, sometimes 1.7. So it's kind of a lottery, I think. So, but then again, you can actually can extract more data out of the flow cell, and that can actually give you a little more value for money. Again, we have actually seen Q40. We haven't seen it Q50 yet, and we are looking forward for the kit that actually they're going to release. And we have done enough number of sequencing to see like this data looks pretty good. So these are our data. Like you can see. So again, you cannot compare Illumina with ABRF in terms of Q values. The reason why that the way the Q values are calculated in Illumina, it's based on a bin value, and here actually it's a base wise value. So those two numbers are not identical. So I will take this as a with a grain of salt. This is my plot, but still I want you to be aware of that caveat. So yes, so it's pretty good amount of data is actually at Q40 range, and then we looked at how much data we need to trim because of the quality. We are seeing quite a low amount of data needs to be trimmed. And if you think about achieving certain X coverage, 15% less sequencing you need from ABRF to cover same amount of data from Illumina. So this is based on our exome sequencing data. So what we have done at Yale, we use both flow cells, like 2x150 as well as 2x300 flow cells for various projects. So now we are actually using quite a bit of this. We successfully sequenced RNA-seq, both ribodepletion and poly libraries, whole genome sequencing, whole exome sequencing, and whole genome methylation libraries on this platform. I have to say one quick thing here, that whole genome methylation library sequencing needs a little more tweaking in terms of when you use adapt chemistry on that. So that needs slightly different kind of treatment compared to others. We also tried both conservative as well as expert mode. We tried elevate, adapt, and adapt plus chemistry. All these three chemistries we have tried for various projects. This is something that we actually was very proud of, like we tried Watchmaker Kit with element adapter and made the library. Because the advantage of doing this is that Watchmaker kits they are also providing us automation possibilities. So just changing the adapter alone, we can actually convert whole Watchmaker pipeline into a VT pipeline for library prep. And this, we have sequenced some of the samples, and I'm going to show you some data. We have sequenced 600 base pair insert sites. We are also looking at using AVT for long reads, long inserts, not long reads, actually long inserts. And so we have even tried 600, and now we are actually pushing the boundary to 2 KB, 3 and 5 KB. And this is important that you can use large insert libraries and sequence as a short read, but that will give you more information about structural variation. So you can actually, instead of sequencing long read, you can actually use short read with long insert to get those information. And that is the purpose why we are doing this. And then, of course, we are using this for, also we have used eDNA amplification sequencing, loop-seq, whole genome sequencing, and TCR-BCR diversity. So we have done actually a variety of different projects on AVT to look at what it can do and what it cannot do. So we also tried loop-seq. Loop-seq is actually, you are generating a long read from the short read platform. You can actually use loop-seq for both AVT as well as in Illumina platform, but we used AVT to do loop-seq. So basically what you're doing, you share your DNA, add a barcode, and then you amplify this after the barcode, and then you allow this barcode to hop. And based on that, you then go back and stitch it back into long reads. That is what you are doing here. And off the shelf, we have the 16S and 18S product available from Element, and then we can also use this for amplicon sequencing and for the applications like looking at targeted sequencing and CRISPR off-target effect and many other similar kind of application. We tried this for 16S data. So we sequenced one microbial data for this, and what we found that we could actually get data up to species level. So when we were using these same samples previously, people were able to get Enterococcus, but they never got Enterococcus facialis. Up to the species level, they could never resolve. They could do that using qPCR, but through sequencing platform, they could not do it. And we have actually able to achieve that level of resolution using loop-seq. So it's very, very, very interesting in that way. We also tested various kinds of library prep like Watchmaker, then we converted Illumina library, then we were using mechanical sharing and enzymatic sharing, and we are looking at how these various approaches works. And so if you look at all the variants called, they look almost like similar. So all this various kind of library prep, it's pretty flexible. This platform is pretty flexible in terms of the kind of library prep methods that we can use. Then we also are now currently using amplicon-based loop-seq. We are doing targeted capture, and then we are also trying sequencing long insert library. Finally, just couple of things that I need to just I want to mention. One, AVT is actually slightly sensitive to loading concentration. It's not slightly, it's actually very sensitive to loading concentration. It took quite a bit for us to stabilize our whole genome and RNA-seq library concentration. Either it goes, immediately it will be under-clustered or over-clustered. And one of the reasons I think that the way the library is quantified is not using qPCR. qPCR method don't work well on this. So essentially it is done through the picogreen or those kind of assays. I don't know how much they are reliable. I think that is where the problem is. But other than that, we are still struggling with large amplicon libraries where if we are using amplicons and then not large amplicon, any amplicons if we are using, it's hard to quantify and so it always need one more effort to sequence, get it done right. And if you are over clustering AVT flow cells, immediately you will see a drop in the Q40 values. So that is something, the Q40 values in these runs are very sensitive with the clustering. So this is one of the caveats that we have. And the other issue is the enzymatic protocols for the AVT somehow pretty variable. So that is why we decided to move into mechanical sharing, and they have a protocol where you can use mechanical sharing for there. With that, I'm just stopping at this point. So this is my group, and thanks.
Awesome. Awesome. Thank you to Kirsten and Bonnie both for speaking on these awesome instruments today. I'm also going to speak today. I'm going to speak on the Illumina NextSeq 2000. And I want to reiterate Bonnie's thank you for bearing with us. I know it's late. And I'm really big on crowd participation, so I'm going to be asking a few questions in a little bit, so be prepared. So first off, I just want to give a little introduction about myself. Why am I qualified to be up here? I work at the University of Wisconsin-Madison Biotechnology Center, and we were founded in 1984, and fun fact, the founder of our Biotech Center is also one of the ABRF founders. So if you've not talked to Ron at all this week, I highly recommend talking to Ron. He's great and lovely. I don't know if he's here today, right now, but anyway. Also, I just want to we do have a few cores within our center. We have DNA sequencing, gene expression, bioinformatics, genome editing, and mass spec. And then if you've never been to Madison, it's a very beautiful town, so I highly recommend come visiting me. Just me, though, nobody else. All right. Also, kind of same as Bonnie, we do have an assortment of instruments. We currently do operate for Illumina 3 MySeqs and NextSeq 2000, and I just realized this is an outdated slide. That is a 6000, that's not an X Plus. We have an X Plus. The 6000 is no longer. Her name was Willa, and she was retired. Our X Plus is named Marcella, and we kind of hold on to this tradition of naming our Illumina instruments after female aviators. I don't know if Marie is in here, but Marie, our fearless ABRF president, she started the tradition because she used to work at the Biotech Center. So we'll get into that a little bit later, though. We also do operate a Promethean P24 and then 2 Sequel 2s. So I kind of want to go over like how do we use the NextSeq 2000? And I'm going to give a little disclaimer here. Our NextSeq 2000 was free to us. So I know you're privileged, Molly. No, for real. Like there was just somebody else on campus who was like, "Hey, we don't want this. We're not using it. My lab is closed." And I'm like, "Oh, sure, we'll take it." So we took it, and I'm not going to lie to you, it was probably a solid 3 months before we actually used it, because we do have the X Plus. So I'm going to be completely honest with you, it's not a heavily used instrument currently, because I think the NextSeq is really a niche instrument. I think it's great, but I think, and I just realized that I did not change the slide. Well, pluses and minuses for both of them. So anyway, this is a very big error, so you can just listen to me. But basically, so the positives we use currently are for 16S and ITS amplicon sequencing. So right now, that's where we find the cost benefit for our projects, because right now the MySeq is great, and the MySeqs are workhorses. We've had them for years, probably some of the earliest serial numbers. And so, but we realize that the quality is actually much better on the NextSeq. So you can kind of see here, and I tried to squeeze a little bit in. On the top, there's the quality of a 2x300 run for our 16S V3, V4 amplicons. And you can see the quality drops very rapidly on the MySeqs. And we were like, that's fine, whatever. Customers never really complained. I mean, because if they do, they're not going to tell me directly. But we then decided we're going to well, the NextSeq has 600 cycle kits. Why don't we run it on the NextSeq? And so we did. And the NextSeq 2000, I would say the first time I saw these runs, I was like, that's all Phi-X, right? That really can't be our amplicons. And it really was. Like the Phi-X was, probably 20, 30%. But this is definitely a much better improvement over the MySeq on running these amplicons. And we do a lot of this metagenomic work. And so for our 16S, this is really beneficial for us to use. So I kind of wanted to just also briefly touch on what I mentioned, the NextSeq is kind of a niche instrument. What I'm saying with that is Illumina's been around. So Illumina chemistry is around. People use it. It's there. And so I think, if you want to stick with something Illumina-based, the NextSeq is great. What helps with that is the NextSeq is very fast. And I don't know if anybody's ever actually run the NextSeq themselves. I don't know if you physically have done it, but it's fast, right? Like you literally just take your tube, you pipette the sample, and you put it in the machine. You're like, woohoo, I'm done. I mean, that's fast. So I'm thinking, clinical assays that need that speed, that turnaround time. And, if it's already prepared, libraries are oftentimes already prepared with Illumina adapters. We don't have to worry about changing out the adapters. And then my next question is kind of, what is your client? what is your client? So what does that client need? And I guess it kind of also goes with what I was just saying with if, their data has already come from Illumina, they want to stay with that route, or, if they need that really fast turnaround time, I oftentimes will throw, if somebody needs something fast, if I can't fit it on our X Plus currently, because obviously that's much cheaper, we'll throw it on our NextSeek 2000. And then kind of going along with the next question is, what is our usability? And it's kind of like, if you can't pipette, you probably shouldn't operate an NextSeek. But the thing is if you learn to pipette, you can operate an NextSeek. So I kind of wanted to make this brief because I really do truly want to actually kind of talk with Kirsten and Bodine so we can kind of get all options. And I'm sure you have all thought of at least 2 questions each, correct? Some nods. Okay, cool. So just a really quick thank you to my team. If you know anybody from Wisconsin, this is my team, and Emily's also in the audience today too. So she is a NextSeek runner. So while you guys are all fighting over the mic to get to your questions, I have 2 things for you. 2. 1, can I get a show of hands of somebody who currently operates a NextSeek 2000? Oh, okay, wow, that was more than I was expecting. All right, how many people operate a G4? Okay, and how many operate an Aviti? Okay, this is a great group. I'm so jazzed for this. Okay, that was much better turnaround than I was thinking. Okay, now my next question is actually going to be for Kirsten and Boney, so we'll start. But I wanted to ask if you guys have names for your instruments, because I think this is the most important question, because I know that we're going to give them time to line up. So.
I would love to name our instruments, but again, Scott Ty is our director of technology.
What do you want to name it? I'll make you do it.
We're still working on that, but yeah. Just be very real close.
For us, actually, we don't have a name for instruments, but we have names for freezers.
Okay.
All the freezers are named after one or other female scientists. So famous females.
Perfect. So ours are named after female aviators. Our NextSeek, her name is Pearl. FYI, she was a kind of a stunt pilot. Probably, I think, one of the youngest stunt pilots. So Eula Pearl Scott, I think, something like that. So yes, we've also had an Amelia and a Sally. Don't worry, they're retired. So does anybody? I'll start with you guys. Otherwise, I do have a list of questions that we're going to get to, but I was wondering if anybody else would like to. Oh, somebody lining up.
You first.
Yeah. Okay. Go for it.
I guess same question for all 3 of you. What are your final loading concentrations derived from? Are you doing qPCR? Are you doing qubit, Evergreen, Cyber, the plate assay?
Yeah.
What's going on?
Yeah, so for us, we do qubit and bioanalyzer. So basically, just figuring out from there.
Yeah, I mean, for most of the cases, we are using qPCR. But if you ask my personal choice, I would actually like something like Normalize to do this, because that actually gives a little more control, but I could not convince my technicians yet on that. So my wishful thinking is that probably something like a Normalize kind of a methodology, which will make it more uniform and less error prone. But the gold standard probably is qPCR, and I will be less inclined towards any other dye-based method for library quantification.
I'm with Kirsten, though. We just do qubit and tape station. I know people are like, yes, qPCR, and I agree. I'm not saying qPCR isn't accurate, but it works for our workflow, and we can base everything off of that, so that's what we do. So I hope that answers. I don't know where you went, but oh, there you go. Hope it answers. Yes.
Scott just texted to send his regards. He says he misses everybody, and Minneapolis is too cold.
He lives
Pretty
in
rich
Vermont.
for somebody
Yeah.
from Vermont.
He'll be at Vegas next year.
He is stoked for Vegas. He did send that as well. So many, many questions, but I'll limit myself, all right? So one of the big deliverables of the ABRF Next Gen Sequencing project was standard data and standard reference materials. So does anybody know of sequencing on these latest platforms using the genome in a bottle reference materials so that we can go back and actually compare head-to-head against previous iterations of kits and machines.
So we have done that. We bought our AVT from a S10 grant. So when we were writing the S10 grant, we used Genome in the Bottle, and we compared that with various platforms. So we actually tried NextSeq to 2000, NovaSeq 6000, and then AVT. I wanted to actually try G4 at that point of time, but nobody ever responded to. So when I was writing the S10, actually I was looking for singular G4. But since I could not get any response, that's where I went to AVT. But I think that's where so I could not make it happen. But we have the data, and unfortunately or fortunately, we never actually published it. But we have that
Great,
benchmark.
yeah, so what would it take to publish it? Do you need a little more, or
No,
do you need the GRG to give you a hand? Or?
So there are two reasons why we did not publish. One, that little bit, being lazy, other things are there, and publication is not something that actually is our priority. And the other issue is that, when you do any kind of a technology evaluation, there's a chance like somebody will blame you for being biased for this reason or that reason. So just simply don't want to be part of that conversation. So we know what the data looks like. And I'm happy to actually share any of this data.
Yeah, so to the first point, definitely share, and maybe a result of this session, you could post it somewhere on a server that the ABRF community could get at. On the second point, I think ABRF would stand behind you to sort of limit your exposure to that, because that's what we do, right? So it may be that we need to add in a few more sites for independence, or it may be that you want some help with authoring it or something like that. But if you back it as part of the GRG study or as part of the next-gen sequencing study, then I think you sort of limit your potential political repercussions.
Thank you.
The data are most likely outdated anyhow.
Yeah, no, definitely.
That's a good point. I don't, Kevin or Kevin, okay.
So when you're running the non-Illumina platforms, what conversations have you been having with your users? Say you're running it on the Element or the SG4, and then you have data that you've been generating on an Illumina platform. How are you continuing those projects that are ongoing on an Illumina platform?
Yeah, so for us, when we were purchasing a new instrument, we put together a purchasing committee of our highest users, so that we could get feedback from them on what they were comfortable with, and we had some users who said, you know what, I'm not interested, I'm not coming back. They had already kind of left our core and were sending their samples off anyways. Yeah, there's that. And we're like, well, we can bring our prices down a whole lot. And they said, oh, they want to sequence on an Illumina machine, and that's fine. But, we do have a lot of users who were like, great, fantastic. And, we did have a few different library preps that we had sequenced in-house on our HiSeq, and we also sequenced them on a G4 as well as an AVT. So we were able to come to them with that data and say, hey, look, this is how they compare. It's all, fruit, right? So our investigators aren't too concerned, in our case at least, with sequencing on a different platform, especially since it's a very similar type of sequencing.
So for us, actually, in AL, there are not just in AL, anywhere else. Actually, there are three types of investigators are there. So somebody who is pretty expert, they know everything about genomics, and they can actually, take apart anything and they can put it back themselves. So, and then there is a second type of investigators who don't know anything about genomics and they want to do genomics. And there's a third one who is in the middle. So the first and second category is easy to convince, because the first one, they know already that, okay, there's a new player in the market, and then they know everything about the machine and the technology and the quality. So you don't have to convince them for anything, and they will automatically come based on their need. The second one category, who don't know anything, they are completely relying on you to make decision. And so for that one, you can actually say, hey, this is advantage, and I think you should use this. So those two categories are easy. And I think the third one, it's like really, it's kind of like I was sequencing all these years through Illumina, so I don't want to change kind of a thing is there. And I think it will take a little more time to say like all these platforms are equal, and it's just like, at this point, actually, I just remember George Orwell, who said like all animals are equal, but some animals are more equal than others. So I think at this point, Illumina is more equal than others, but it will catch up.
I had a question for the Aviti, because it can do the 2x300. So Molly showed really good data for quality scores on amplicon-based libraries. Have you guys looked at comparing like MySeq 2x300s to the Aviti 2x300 amplicons?
So if you are comparing between 2000 and Aviti, data is pretty comparable. But I have to make one more point on that. So in 600 cycle kits, we are seeing a rapid drop in Q40 scores at the end of the read, almost like last 100 base where we see a very quick drop. So we contacted Element, and they gave us a new upgrade, and we thought that with the new upgrade, they told us that this will be solved. But the last run after the upgrade, we are still seeing the drop. But then the problem is that that particular run was over clustered also. So we were expecting 500 million reads, and we end up having around 300 million reads. We have got like 100 million read more in that. So, and that is another challenge that we are facing with amplicon sequencing in Aviti, that we are yet to get a handle how to actually find real loading concentration, so that the run is not under clustered or over clustered. And as I said, like, the moment it's over clustered, the quality drops fast. So we don't know at this point that whether the drop in the quality at the end is because of, and especially R2 reads are actually suffering most. So we don't know whether is it because of a technical issue, or is it because of the overloading. So we are rerunning that library. Hopefully we will have some answers regarding that.
Right. Oh. Go ahead, Sarah.
Yes, so my question is generally about the folks who've done the same library type, both as a de novo and a converted library. Have you seen a appreciable difference in the data quality in terms of PCR duplicates, if it has a PCR, or quality scores, et cetera, between the de novo versus the converted libraries for either platform, Aviti and Singular?
So one of the major challenge we had with the conversion libraries that in Adapt Plus, the loading concentration is not same as what it is. So we struggled a bit with Adapt Plus to Adapt and Adapt Plus libraries to figure out what is the real concentration. So loading concentration will elevate, and the way the library works on the flow cell is slightly different. So that is the one big difference we have seen. But otherwise, I don't think that there is any big difference in terms of data quality.
Yeah, we haven't really seen much different. Like, we're obviously concerned with, it's the conversion for us is it's a PCR conversion, right? So we're like, oh god, we're going to introduce all these duplicates, and it really hasn't been that bad. You put in enough, and we try to do as few amplifications as we can get away with. So we haven't really seen, an effect there. I will say there's been one library type that we have not been able to convert. Or Singular was able to convert it, but it wasn't great as far as comparing the de novo versus the converted, and that's the GMX libraries. Those have been kind of an interesting puzzle for us.
Oh, yeah. I use an X6, so I don't convert. I think Graham, you were.
That was kind of my question, but it brings up another one. Do you envision that we'll eventually develop different parallel oligo systems, or are we always just going to be using Nextera and TrueSeq, and then there's always a conversion if we want to engage these other instruments? Do you think manufacturers of kits are going to start doing 3 parallel oligo systems?
I mean, we would love to see that, right? So, I mentioned in my talk, we were working with RhinoSeq, and we were working with RhinoSeq to build a kitted library prep that includes Singular adapters. Because a lot of these we are just like, we get the kit, and then we go get adapters from Singular, right? If we want to do a de novo prep. So, I think it takes us pushing the vendors and our partners saying, hey, this would be awesome, this would be amazing, because right now, our users, they're just like, I'm gonna buy my Illumina adapters, and you guys can deal with the conversion, which we're okay with, but.
So we looked at the watchmaker protocols, and now we are actually, just by replacing adapters from Illumina to Element adapters, we are able to make libraries and we are able to sequence them. But with AVITI, there's something new, that they have a freestyle kit coming up for sequencing, which makes like all this conversation about the adapters irrelevant, because then end of the day you can use anything you want. And the advantage of those, like we were trying with this environmental DNA, there are almost 284 samples we have to prep library and then pull them together to run on AVITI. So AVITI don't have that many indexes. So we used Illumina indexes, and then we pulled everything and then converted and then run on an AVITI instrument. So that way we are able to use the advantage of both the world. Like whatever Illumina's all these years of the R&D that they have done, whatever they created, we are essentially actually using that for that advantage of that into the AVITI. So I think having this kind of three different scenarios, probably if you can actually use it for your own strength, probably it's more beneficial rather than generating a common like SOP kind of a thing for all the platforms.
Yeah, hi. First of all, thank you for doing this. Very, very useful. I just wanted to mention, because it was, I think there were some questions about conversion and how it affects duplicates and all these. There will be data tomorrow at 3 at the GRG project update session. Data and slides and analysis. So if anybody has questions about that, shameless plug, come tomorrow.
What time and what location?
3 and GRG study update is the name of the session. Yeah, I don't know the location.
It's probably
follow Catherine.
Right,
Just
it's
follow
probably in
Kathy
one of these
where
rooms.
she goes.
One clarification, one question. Have you at Yale played with the freestyle flow cells yet?
No, not yet.
Okay, because that was an answer to one of the previous questions. He was asking about the adapters fitting. Do we have to convert? Element just released flow cells that will take Illumina adapters native now, circularized on board, so we don't have to convert anymore. I haven't run them yet either. I'm getting my first one this week, so I don't know how well they work. We're going to find out. And then the other question that was asked, I forget, it was this lady here. I have one user, count it one, that wanted to know how the 10x libraries would compare Illumina versus Element. So she took the exact same library, had me run it on Element, sent it back to Ohio State where she had come from, had them run it on Illumina, and then had their informaticist overlay the data sets, and the informaticist says, yeah, it's beautiful, it matches perfectly, no worries, go. So just a clarification point.
That's awesome. Thank you for sharing. Does somebody have I feel like you guys are just, go for it. Yeah.
There was some mention about library sizes affecting some loading concentrations and yields. What have you guys seen on the 3 platforms in terms of smaller libraries, bigger libraries, and how does it affect loading concentrations?
Yeah, that's where we are struggling with. So if the general recommendation that actually Element gave is actually pretty straightforward, but the instrument is not actually behaving to that concentration. So it needs a little more, I think we need a little more time and experience to really understand that what is that relationship is. So at this point, at least we know that for a whole genome sequencing, we are comfortable. So if library size, average like whole genome libraries are around 320, 340 base pair median size. So those libraries, now we know around 8 to 10 picomoles of loading is actually perfect. But we are not yet able to identify those things with respect to amplicon libraries and as well as, and the other thing I think when this conversion is taking place, I think it was also actually, in my understanding, whatever is happening, like now they have onboard conversion and all those things. So there is some level of differences in efficiency in each run. Probably that is what affecting more, and that might be the reason for why we have under-clustering and over-clustering. Because the amount of differences that we are seeing is actually not really reflective of the concentration. So I think something in between is actually, and that's why I think like freestyle probably is going to take care of this, and we will do better with that freestyle kit. So I'm just kind of waiting for that to see like if my intuition is right in that.
And so that makes me think of another question. When you're looking at conversions on both singular and an element, have you guys gone to compare what you're seeing on your traces to what you're seeing bioinformatically for the insert sizes?
Yeah, we haven't done that per se, but at the same time, as far as the conversions go, our traces look very similar from like the library that we've received from our user to what we get back and throw back on the BioA after the conversion. So, you know, similar looking fragments.
So same goes to AVT, that the traces looks, and that was the last slide that I did not actually explain well. So yeah, the traces look same, but the only issue is that when we looked at 2000 base insert size, then the normal protocol, if you're running normal sequencing protocol over that, it all of a sudden then it will actually shift the median size to 250. So now, so basically what is happening, the instrument is now started favoring the lower fragment size instead of larger. So there is a new protocol which Element shared with us. After implementing that, actually now we can see the real. So if you are going beyond up to 600, we have seen that it perfectly follows whatever your traces are. And if you are going for longer insert than that, then you have to use a special protocol for sequencing, and that otherwise it start favoring going other way around.
Okay, I do have another question for well, all of us. How is the technical support?
Okay, so there are two things here. Actually, I think, and I think that is a key aspect of deciding that whether you want to buy a particular mid-throughput instrument. And first thing is, of course, technical support. And AVT so far, they were pretty proactive, and they always, respond quickly. The other aspect I think also you should also think about is the service contract. The reason why these are mid-throughput instruments, you can only run so many samples or so many projects. And if you are a core facility like Yale, which is based on cost recovery model, that means you have to spread your service contract into those number of samples. So you are left with only very few number of samples to spread that cost. So service contract should be also, if you think from the technology perspective, all these platforms are more or less equal. I mean, we can argue this academically, that this is better than that, and we can put some numbers, some graphs, some plots. But eventually, when you think from a core perspective, the real question is how much every year we have to pay for service contract, how much is per gig cost is, and then how good is a support. And support is most critical because some of the companies, those who are actually into this field, like AVT, Element, or Singular, they are relatively new, and they are smaller companies. So we don't know how long they are going to be in the market, how well they will be invested in their customer support. So these are some of the most important things that we should definitely think before buying a mid-throughput sequencer. It's not like, unlike old days when we have only one system and we can't actually make any decision because whatever is said, we have to do that. So yeah, so far AVT, we are very much happy with their customer support. They were amazing.
Any vendors that are here, I want to reiterate, yes, we have to think about service contracts. Let's bring that price down, yeah?
Yeah.
Sorry.
Please, right?
Yes.
We were a little aggressive in our negotiating on service contracts. But no, that was definitely, it might have been in the intangibles part of our considerations, but that was nerve-wracking for us, right? Because we were very seriously considering unknown companies and unknown customer support and unknown technical support. And this is our sequencer pretty much. And we knew Illumina, and we know that if we call, somebody will pick up. And usually people don't like to come up to Burlington, Vermont. It's a haul, but we would have an engineer there. We have had fantastic customer support with Singular. Again, people don't like to come up to Burlington, Vermont, but we've had same day, next day engineers arriving, and they get back to us quickly if we have a problem that can be solved over the phone or internet.
Oh, go for it.
Knowing what you know now, would you choose the same sequencer again?
There are vendors in the room, right?
I mean, I think yes, and as I said, like, all these instruments are fabulous. They have their own place, they have own niche market space in the sequencing market. So I think the question is that if I buy another AVITI, if I have to buy another mid-throughput, probably I will look for a little more diversity, because I would like to add singular, because that also comes with its own thing. But doesn't mean that I don't like AVITI. I like that instrument a lot. The reason only that I want to buy a second one, because that comes with its own strengths, and I want to use that capabilities also within the core. So yeah, I will go for AVITI.
I will actually second that. We always say, if only we could have 2 sequencers, right? We'd probably get an element. They do fill different niches for us, right? So right now, at least, Singular can't do Paradigm 300. And we do love metagenomics. So, we do still have our MySeq, and that's what we're running if we need to do really and truly a Paradigm 250. But, yeah.
And then I would like to say, while yes, I did get my NextSeq for free, I think we are and will be interested in a lot of the new technologies that are coming out with Singular and Element with the spatial sequencing. So we're interested. We also have to pay for it this time around, so. Okay, any other, we do have a few minutes left if anybody wants. Oh, Sean, back again.
How long did it take to get the instrument up, running, your assays validated, and your services running smoothly for each of the instruments?
I think for us, actually, it's 2 weeks' time we could do that. The engineers came, they set everything up, and then. But if you are really thinking about giving a reliable service, then I think it needs a little more time, because you need to actually really play around with your loading concentration, learn, to play with the library types, and so you need a little more time. You have to invest a little bit to get it. But if you're just thinking about, ready to use system, then it's like 2 weeks time, and we were pretty much able to use our system from then onwards. And as I said, element support is pretty good. So technical people, they, as well as the FAS, and everybody was always hands-on. So you email, and no emails takes more than an hour to get replies. So it's pretty good.
Yeah, we were up and running within 2 weeks. We kind of flew by the seat of our pants. We're just going to sequence stuff, right? We were pretty excited, since we hadn't really gotten to sequence much, because our HiSeq was end of life, so had been for a few months.
Yuri?
To Molly's question, we do name our instruments. One
Yes.
of them was making sound, the other was not, so obviously it's Jay and Silent Bob. But my question is, so nobody even mentioned complete genomics, BGI, MGI, so it's not a thing anymore? Or?
So that's a great question, and as session organizer, I will definitely answer that question. So I went based off of my network, and I currently don't know anybody. I also asked around. I also asked for the PacBio Onso, which I don't know anybody in the network. So I did ask for more, and I'm not slacking on my session organizer duties, but I didn't know, and I mean, I think my network is relatively large. We've got the Secret Science Club going on, but I asked around and I didn't get any of that feedback, so I'm not saying that they don't exist or they're not important. These are just the willing people who wanted to come sit up here with me. So that's a great question, Yuri. I would love to know, and I think maybe we could have a follow-up that maybe includes them. So,
one thing I can definitely tell, Yale, one of the places in Yale, they got a what they call G4 or something, T4 or G4 or something. It's basically a mid-throughput sequencer. The Stem Cell Institute got it. So data is pretty good from that instrument. There's no doubt in that. And the cost for running that instrument is also low. But our nervousness was about the legal side of it. And so most of the projects that we are running on an NIH grant, so if suppose we invest on an instrument and then all of a sudden this legislation, which is kind of like we don't know where it is going to be. So if it comes into play, that means like we can't sequence anything, because basically most of the sequence that we get is coming from NIH or NSF grant. So only like less than a fraction, less than 1% sequencing comes from the private companies, and that is the only one probably we could be able to do that. So I think it has nothing to do with the quality of the sequencing or the quality of instrument. They have a good support team. I know a couple of guys who actually spoke to them many times. So the issue is this uncertain political environment, and we don't know how to navigate that.
This was a great conversation. I'm so happy you guys all came. I do have one small request. Can we clap very loudly just because I want people to know this was a really great session?